Welcome to Bachelor of Science 3rd year, subject Botany, the 5th semester, course code BOC 107, course title Microbiology and Plant Pathology. This is Unit 10, Genetics of Pathogenicity. The name of the module is Genes for Virulence and Avirulence and their role in susceptibility and resistance. I am Ms. Marina Janice Rodriguez, Assistant Professor in Kamal College of Arts, Science and Commerce for Women, Nove, Goa. In this module, we will be seeing the introduction, virulence and avirulence, gene for gene concept, avirulence genes and other genes involved in pathogenesis and virulence by microorganisms. By the end of this module, you will be able to explain the concept of virulence and avirulence, define the gene for gene concept, state the characteristics of avirulence genes and its role in pathogenicity and virulence. You will be able to describe other genes involved in the pathogenesis and virulence by microorganisms. Know that infectious plant diseases are a result of interaction of at least two organisms, the host plant and the pathogen. The properties of each of these two organisms are governed or controlled by their genetic material. It has been known for more than a century that the degree of susceptibility in plants or resistance to various pathogens is an inherited characteristic. Consider this particular situation. One plant variety inoculated with two different strains of pathogens. The plant variety is susceptible to one race of pathogen but is resistant to the other. One pathogenic race possesses a genetic characteristic that enables it to attack the plant while the other race does not. Let us consider another situation wherein we take different plants and infect them with the same pathogen. So, the same race of pathogen is inoculated on two appropriately chosen varieties of host plants. One plant variety is susceptible while the other is resistant to the same pathogen. This means one variety possesses a genetic characteristic that enables it to defend itself against the pathogen so that it remains resistant, while the other variety does not. Virulence and avirulence. It is noted that one pathogen race can infect a certain group of plant varieties, while another race can infect another group of plant varieties, including some that can and some that cannot be infected by the previous race and so on. Studies of the inheritance of resistance versus susceptibility in plants prove that a single gene controlled resistance and their absence would allow the plant to be susceptible. Similarly, similar studies of inheritance of avirulence versus virulence in pathogens prove that single genes control avirulence and their absence would allow virulence characteristic in the pathogen. The ability of pathogens to inherit their infection type, that is the degree of pathogen virulence or avirulence, has been studied intensively only since the 1940s. It is now clear that the pathogens consist of a multitude of races, each different from the others in its ability to attack certain varieties of plant species but not the other varieties. Studies of their interactions prove that R genes in the plant are specific for the AVR genes in the pathogens. Thus, varieties possessing certain genes for resistance react differently against the various pathogen races and their genes for avirulence. The progeny of plant varieties react to the same pathogen in exactly the same manner as the parent plants. This indicates that the property of resistance or susceptibility by the plant against a pathogen is genetically controlled, or we can say that the property of being resistant or being susceptible to a particular plant pathogen is inherited from the parent plant by the progeny. Similarly, the progeny of each pathogen causes on each variety the same effect that was caused by the parent pathogen, indicating that the property of virulence or avirulence of the pathogen on a particular variety of plant is also genetically controlled, or we can say it is inherited. Thus, under favorable environmental conditions, the infection, that is the susceptibility of the plant, or non-infection, that is the resistance of the plant, in each host pathogen combination is predetermined by the genetic material of the host and of the pathogen. The number of genes determining resistance or susceptibility varies from plant to plant, as the number of genes determining the virulence or avirulence varies from pathogen to pathogen. Coming to gene for gene concept. Gene for gene concept explains that for each gene that confers virulence to the pathogen, there is a corresponding gene in the host, that is in the plant, that confers resistance to the host and vice versa. Resistance through hypersensitive response has been shown to be a result of a gene for gene system in which a avirulence gene, that is the AVR gene in the pathogen, will correspond to a resistance gene in the host plant. So that means if the host plant is resistant to a particular pathogen, that means that particular pathogen is having a corresponding avirulence gene in it, which is not allowing the pathogen to cause the disease in the host plant. It is a result of the plant specific signal molecules, the elicitors, produced by the avirulence gene of the pathogen and recognized by the R gene coded specific receptor molecules in the plant. Coming to properties or characteristics of avirulence gene, also abbreviated as AVR genes. They were first identified by H. Flor in 1950s. The AVR gene makes the pathogen unable to induce disease, the avirulent for specific activity of the host plant. The AVR genes determined the host range of the pathogen at the species and at the race or even the variety level. As the gene for gene concept implies, in the majority dominant resistance, that is the R gene, in the resistant host corresponds to a single AVR gene. The AVR gene also determines not only the plant species but also the cultivar the pathogen can attack. Role of AVR genes in pathogenicity and virulence. Most AVR genes tested so far play no role in pathogenicity or virulence of the pathogen, since even when AVR genes are inactivated by mutation, susceptible host continues to be susceptible. However, the AVR gene BS2 gene from Bacterium Xanthomonas campestris pathovar vesicatoria encode proteins that are also necessary for pathogenicity. The AVR gene is present in all strains of this pathogen, whereas the mutants lacking the AVR gene loses pathogenicity in all the susceptible hosts. Other genes involved in pathogenesis and avirulence by microorganisms. Irrespective of plant infecting pathogen possesses the AVR or the HRP genes that may determine their pathogenicity on certain species or cultivars of plants, they are likely to possess one or several additional classes of genes that are essential for the pathogen to cause disease. These are called as pathogenicity factors. Other classes of genes for the pathogen to express increased virulence on only one or a few related hosts are called as virulence factors. Pathogenicity factors encoded by pathogenicity genes, also called as PAT, and disease specific genes DSP, are involved in steps very important for the establishment of the disease. These include those which are essential for attachment of the pathogen to the plant surface, germination and formation of infection structures on the plant surface, penetration of the host and colonization of the host tissue. These are my references. Thank you.